Students, we are aware that the human body consists of several organ systems. If you have any idea, cells are the structural and functional units of the body. Cells of any one type form a tissue. Several tissues come together to form an organ, and several organs come together to form an organ system. Each organ system performs a particular physiological function. For example, say digestion. Digestion takes place in the alimentary canal, which is an organ. Associated with this alimentary canal are several organs like liver and pancreas, as a result of which alimentary canal is able to digest the food, and this digested food is absorbed into the blood. Now, in the body, thus each organ system performs a particular physiological function, whether it is digestion or excretion or say respiration, and overall there is the nervous system to control the functions of all these physiological functions and the organ systems that are responsible for these physiological functions. Now, in the body, there are two types of control for the control and coordination of control as well as coordination of the different organ systems. One is called the neural control, the other is called the endocrine control. Neural control is the control or coordination exercised by the nerves that arise from the neurons or groups of neurons that are located either in the brain or in the spinal cord. And this kind of control is a rapid control. The other type of control that is called the endocrine control is exercised by the hormones that are secreted by the endocrine glands. These endocrine glands are located in strategic sites of the body, and their secretions are called hormones. These hormones are secreted directly into the blood, and that is why they are richly supplied with blood vessels. Blood then carries these hormones to their target organs. Every hormone has a target organ where it exercises its control and coordination activity. Now, endocrine control is somewhat a slow type of control, as the neural control is a rapid control. This is a slow control. Now, coming to the neural control or control by nerves that arise from the brain and the spinal cord from groups of neurons located in them. So, the structural functional units of the brain or the spinal cord or the nervous system are the neurons or nerve cells. These are about 10 to the power 12 in number, 10 to the power 12 in number and that means there is a very large number of these neurons. And the peculiarity and the interesting features of these neurons is that they are post-mitotic cells. So 10 to the power 12 neurons, and these are post-mitotic in nature. That means after birth, there is no increase in their number. So you will ask me, if there is no increase in number, increase in number is called hyperplasia. Increase in number is called hyperplasia. So there is no increase in number or hyperplasia after birth. There is only hypertrophy. That means there is a second kind of growth. Number doesn't increase, but sizes of these individual cells, the neurons, increases. So there is hypertrophy. So there are two kinds of growth therefore in cells. One is hyperplasia or increase in number, and the other is increase in size. And neurons have the second kind of increase in size. And the neurons have the second kind of growth, that is hypertrophy. Another interesting feature of these neurons is that not only their number doesn't increase after birth, their number in fact decreases. That means there is neuronal loss. There is no increase in number, there is decrease in number, in other words, that is called neuronal loss that occurs with age. With age, the number of neurons decreases. And to this category of post-mitotic cells also belong the muscles. So neurons and muscles belong to the category of post-mitotic cells, in which there is no hyperplasia after birth, there is only hypertrophy, and in fact there is neuronal loss. So afterwards there is only neuronal loss, and any loss of this number either in the neurons or in the muscles is irreparable. That means the loss cannot be made up. Now next we come to the structure of the neuron. Structure of the neuron, as you can see in the diagram that is attached to the whiteboard, structure of neurons. First of all, the size. Sizes of neurons varies from 5 to 100 microns. They show a great variation in size from 5 to 100 microns in size. And if you look at their structure, in this top diagram, this shows the structure of a neuron. There is a cell body containing all the intracellular organelles like the nucleus, the endoplasmic reticulum, the mitochondria, lysosomes, and so on. So these are the intracellular organelles that are ordinarily present in any cell, are also present here. And another distinguishing feature is the presence in the neurons of several fibers. In fact, there are two types of fibers. Now, one is large in number and branched. Large in number and branched, they are called dendrites. So dendrites are large in number and they are much branched. Large in number as well as they are branched. And the other type of fiber present in the cell body, this is the cell body, and the other type of fiber present in the cell body is called axon. Now, what I mean to say is that in the cell body of a neuron, there can be many dendrites, but usually there is only one very long, relatively unbranched fiber that is called the axon. Now, what is the function of these dendrites and axons? Their function is to conduct impulses in the form of electrical events. We shall discuss this later. So, they conduct impulses from one neuron to another. Dendrites are called afferent, AFF, afferent. Afferent means towards. They conduct impulses towards the cell body. They conduct impulses towards the cell body, and axon, usually of which there is one very long, relatively unbranched axon, is efferent. Efferent means away from. So, this is afferent or towards, and this is efferent away from the cell body. And with the help of these dendrites and axons, neurons conduct impulses from one nerve cell to another. Now, one neuron makes a synaptic joint with another dendrite. Usually, an axon carries carries impulses away from the cell body, and this carries impulses towards the cell body. And axons that carry impulses away from the cell body make synapses usually with another dendrite. So, such a kind of synaptic connection, actually there is no connection, there is a synaptic gap of about 200 angstroms. That point is called, where they come together, is called a synapse, and there is a synaptic cleft of about 200 angstroms, where information is exchanged from the axon of one neuron to the dendrite of another neuron. So, this synapse may be, usually it is exodendritic, but it can also be exosomatic. That means, directly also it can conduct impulses from the axon to the cell body. So, it may be exodendritic normally, it can also be exosomatic. Now, inside the cell body, there are what are known as Nissel's granules. Nissel's granules, these Nissel's granules, Franz Nissel, who for the first time reported their German histologist, who for the first time reported their presence in the neurons, Nissel, Nissl, Nissel's granules. Now, these Nissel's granules contain rough ER, rough endoplasmic, they contain rough ER. ER, as you know, is responsible for protein synthesis, rough ER particularly. So, they are protein synthesizing particles inside the cell body. They are called Nissel's granules, characteristic feature of the neurons. Another characteristic feature is the presence of lipofuscin granules. Lipofuscin granules. Now, these lipofuscin granules are in a way waste products that cannot be excreted, and they actually remain inside the cell body, and as a result of the accumulation of these lipofuscin granules that are derived from lipid metabolism, there is a loss in the function of the neurons as it occurs with age. So, with age there is gradual accumulation of these lipofuscin granules in the cell body, which affects their functioning. Now, what we call a nerve is actually a bundle of such axonic fibers, arising from a group of neurons in a particular part of the nervous system, be it the brain or the spinal cord. Now, in the second diagram, you can see, before that, now this is the axon. This axon in mammalian neurons possesses a myelin sheath. Myelin is a fatty kind of material, and this myelin sheath is interrupted at places. So, myelin sheath, these axons have a myelin sheath. Myelin is a fatty material of this is present around the axons, in mammalian axons particularly, and this myelin sheath is not continuous. It is interrupted at places by what are known as the nodes of Ranvier. So, this mammalian axons are characterized by the presence of a myelin sheath and nodes of Ranvier in the axons, as a result of which conduction of nerve impulses in the mammalian axons is much faster. Normally, this electrical event that spreads along the fiber during the conduction of the impulse does not have to pass along the whole length of the axon. It can jump simply from one node of Ranvier to another. So, the myelin sheath therefore, in a way, they function just as they function like an insulating material. They prevent loss of electric charge, and not only that, the nodes of Ranvier help to transmit impulses much faster in comparison with the non-myelinated fibers. Now, another interesting you see, this is the before we go to glial cells, let us spend some time on this synapse. So, this is that axon. You see, axon is written here. The axon ending is enlarged to form a synaptic knob. A knob is formed, and this synaptic knob contains many rounded vesicles and several mitochondria. What do these vesicles contain? The vesicles contain a chemical transmitter that is called acetylcholine. Not only acetylcholine, several other kinds of substances are also they also help to transmit impulses across the synaptic cleft. So, the crossing of the impulse along this synaptic cleft occurs is chemical. That means, it takes the help of acetylcholine to cross the synaptic cleft and then bind itself to the dendritic. So, this is the subsynaptic membrane, that is part of a dendrite. So, here it is the passage of the impulse occurs from the axon to the dendrite. So, such a kind of synapse is called exodendritic, and this can be also directly take place on the cell body. So, that will be in that case known as exosomatic. Now, next we come to glial cells. Now, what are these glial cells? Now, apart from neurons in the nervous system, in the brain and the spinal cord, there are non-nervous glial cells. Now, these glial cells are normally of four kinds, and their number is more than that of the neurons. We have said that the number of neurons is about 10 to the power 12, but this number of glial cells in the brain and the spinal cord is much more than that of the neurons. Four kinds. One is called oligodendroglia. These are non-nervous. That means they do not help in the conduction of nerve impulses. These are non-nervous. Non-nervous means they do not conduct nerve impulses, but they perform other important functions. One is this oligodendroglia, which synthesizes this myelin sheath inside the central nervous system. Another, its equivalent is squam cell, which also functions to synthesize myelin, but squam cells are present outside the central nervous system. Oligodendroglia synthesize myelin sheath inside the CNS, squam cells outside the CNS. So, oligodendroglia and squam cells, they are a kind of glial cells. Second is the astrocytes. Astrocytes are of two types, protoplasmic and fibrous. And these astrocytes are so named because of their star shape, and they function as a packing tissue. They serve to store food materials, and as and when required, they will supply the nutrients to these neurons. So, astrocytes. And thirdly, what are known as, there are ependymal cells. Ependymal cells. Now, these ependymal cells secrete cerebrospinal fluid. If you have any idea, brain is not a solid structure, it is a hollow structure, it is fluid filled, and the kind of fluid that is present inside these cavities of the brain, they are called ventricles. They are different from the ventricles of the heart, but these are also called ventricles. So, ventricles of the brain, they are filled with this cerebrospinal fluid secreted by ependymal cells. That means these ventricles in the brain, the cavities inside the brain, are lined by these ependymal cells, and they secrete the cerebrospinal fluid. Another type is microglia. Big micro because of the small size. These microglia are a kind of glial cells that are phagocytic in function. Phagocytic means they are protected. They protect the neurons by their phagocytic action. So, microglia are phagocytic in nature. So, there are thus four kinds of glial cells about which many of us are not aware. We say that the nervous system is only formed of neurons. No, there are also other non-nervous cells called glial cells. They also perform equally important functions, though they are non-nervous. Then we come to the organization of the nervous system. Organization of organization of the nervous system. Now, the nervous system, for the purposes of study, structure and function as well, is divided into two parts: central nervous system or CNS for short, and the other is the peripheral nervous system or PNS for short. This is central nervous system and this is peripheral nervous system. Central nervous system comprises the brain and the spinal cord. If you have any idea, brain is the most vital organ in the body, lies safely protected within the brain box or skull of the head, and spinal cord similarly is safely protected by the vertebral column through which it passes. So, the brain and spinal cord comprise the central nervous system. Peripheral nervous system has two parts. One is called the somatic nervous system, and the other is the visceral nervous system. Visceral nervous system, also called autonomic nervous system. Autonomic nervous system. Somatic nervous system consists of 12 pair of cranial nerves, 12 pair, and spinal nerves 31 pair. Now, these nerves are all paired. The cranial nerves arise from the brain, and spinal nerves, as the name indicates, arise from the spinal cord. While cranial nerves are sensory, motor, they can be sensory, they can be motor, they can be mixed. I will explain them while we discuss their controlling functions. And sensory because they arise from sense organs, motor because they penetrate muscles, controlling their contraction, and mixed means both they are sensory as well as motor. Whereas spinal nerves are almost all are mixed nerves. Mixed nerve means they are both sensory as well as motor. So, 12 pair of cranial nerves and 31 pair of somatic nerves comprise the somatic nervous system. Visceral nervous system or autonomic nervous system has similarly two divisions. One is the sympathetic division, and the other is the parasympathetic division. Sympathetic division and the other is the parasympathetic division. Now, this autonomic nervous system or the visceral nervous system and their two divisions they are responsible for controlling the functions of most of the internal organs which are not under the direct control of their will. That means, if we want, we cannot directly control their controlling function. So that is why they are called autonomic. That means they are autonomous that means they are not dependent on the central nervous system. But shortly again we will see that this is not very true. Anyway, so what I mean to say is that every internal organ which is not under the direct control of our will receives both these fibers, sympathetic, so internal organs which are not under the direct control of our will receive fibers of both these divisions, sympathetic as well as parasympathetic. Usually they have opposite functions. That means, if the sympathetic, for example, in the heart receives both these kinds of fibers, whereas sympathetic fibers have a stimulatory action on the heartbeat, whereas parasympathetic fibers have an inhibitory influence on the heart. Similarly, on the digestive glands, say, the roles are reversed. That means, it is not to say that sympathetic fibers are always stimulatory or the parasympathetic fibers are always inhibitory. Their function depends on the internal organ they innervate. Next we come to the meninges. Another important part of our discussion is the meninges. What are these meninges? Meninges are three protective coverings that are present around the central nervous system. Around the central nervous system, that is the brain and the spinal cord, there are several protective functions. There are several, three in fact, not several. There are three protective coverings. They are called meninges. And what are these? What are their names? The meninges are from inside to outside. That means, inside one is present immediately enveloping the brain, that is pia mater. Then in the middle there is arachnoid matter, and outside there is dura mater. Pia mater is almost thin single layered pia mater, and this innermost covering of the brain or the spinal cord is joined to the brain at two places. The two places are called the anterior choroid plexus and the posterior choroid plexus. So at these 2 points in the brain, this pia mater is attached, otherwise it is separate. Only at 2 places it is attached to the brain. Then there is the arachnoid matter containing an arachnoid space, and finally on the outside there is the dura mater, which is very tough and fibrous, and its function is to give protection to the otherwise very soft and most important brain and the spinal cord. So these are meninges. Next we come to the structure and function of the brain. Structure and function of the brain after brain, will, if time permits, will go to the spinal cord. So, now the brain, for the purposes of study, is divided into three parts. The three parts are prosencephalon, three parts, prosencephalon or forebrain, then there is the midbrain or mesencephalon or midbrain, and there is the hindbrain or rhombencephalon. Now, each of these three main parts of the brain again has subdivisions. Now, the forebrain or prosencephalon is formed of the rhinencephalon, comprising a pair of olfactory lobes. You must have some idea of the function of the olfactory lobes. These olfactory lobes are those that are concerned with the sense of smell. So, their fibers originate from the nasal epithelium, and then they reach out to these olfactory lobes in the forebrain. So, rhinencephalon, they are usually solid bulbs. They do not contain ventricles, they are solid. So, prosencephalon or forebrain consists of rhinencephalon. The most important part is the cerebral hemispheres that form what is known as the telencephalon or cerebral hemispheres. So, the dominant parts of the brain are these cerebral hemispheres. Whatever is shown in this diagram actually is or are the cerebral hemispheres. So, they are also called together cerebrum. So, cerebral hemispheres or cerebrum, which is the largest part of the brain, Then we have the diencephalon. The upper part is diencephalon. Upper part is called epithalamus, and the lower part is called the hypothalamus. Hypo meaning below, hypothalamus. So, epithalamus and hypothalamus together constitute the diencephalon. Then, so this interesting feature of this epithalamus is that this epithalamus, that means the upper part of the diencephalon, is connected to a pineal body. It is connected to a pineal body. Have you heard of this pineal body or pineal organ? That is to which this epithalamus is connected by a stalk. Now, this pineal body is sensory to light, and it secretes, depending upon the amount of light it receives, it secretes melatonin. Now, this amount of melatonin that the pineal body synthesized from the pineal body, melatonin, and this melatonin controls the sleep patterns. That means, actually, this pineal body that secretes the melatonin. Melatonin is inhibited by light. That means the more the body receives light, the more light the pineal body receives, less melatonin is secreted, and this less secretion of melatonin means less sleep. So the amount of sleep is controlled by melatonin. Amount of melatonin that is secreted depends on the amount of the light that the pineal body receives. And light in fact inhibits the synthesis, the secretion of melatonin. Similarly, hypothalamus. From the hypothalamus, which is present on the ventral side of this diencephalon, from the hypothalamus arises the pituitary body. Just as from the epithalamus arises the pineal body, from the hypothalamus arises the pituitary body. And if you have any idea, this pituitary body is known as the master endocrine gland in the body. Master endocrine gland. It consists of, so this arises, so this is like this. This is the, it has 3 lobes, anterior, middle and posterior lobe. So this is the rough structure of the pituitary body, and together these 3 lobes secrete a number of important hormones that have controlling functions on almost all organs in the body. That is why the pituitary body is known as master endocrine gland. And so from the epithalamus arises the pineal body, from the hypothalamus arises the pituitary body. Now, hypothalamus, if you have any idea, is a very important part of the brain. Information concerned with the well-being of the body, all information concerned with the well-being of the body ultimately reaches hypothalamus. And depending upon the information that it receives, it controls the secretion of hormones from the pituitary body. In the hypothalamus, there is also the feeding center. The amount of food that we take in is controlled by the feeding center of the hypothalamus. There is also a temperature control center, osmotic control center. So several important. Come to hindbrain or rhombencephalon. Midbrain or mesencephalon. Forebrain, then midbrain and hindbrain. After forebrain, we come to midbrain. Now, this midbrain consists of 2 pair of colliculi, upper colliculi, a pair of upper colliculi, and a pair of lower colliculi. Now, upper colliculi control vision, whereas lower colliculi control hearing. That means there are 2 bulgings, 2 upper bulgings and 2 lower bulgings. Upper bulgings are called upper colliculi, they control vision. Lower pair of bulgings, they are called lower colliculi, and they are concerned with hearing. And upper and lower, so together they constitute therefore 4 bulgings, or what is known as corpora quadrigemina. And upper and lower of any one side it has two sides. So, any one side known as corpora bigemina. Then we come to the hindbrain. Now, hindbrain consists of three parts: cerebellum, hindbrain consists of three parts: hindbrain or rhombencephalon, three parts: cerebellum, which is the second largest part of the brain, cerebellum, after cerebral hemispheres, cerebellum, that consists of a central vermis, central lobe that is called vermis, and on the two sides we have two cerebellar hemispheres. So, altogether, so the cerebellum has three lobes. There is a central part called vermis, and there are two cerebellar hemispheres. And cerebellum, and then there is what is known as pons. Pons is actually a bridge. Pons means a bridge. It forms a bridge between medulla and the higher centers of the brain located in the cerebral hemispheres. So, cerebellum, pons, and then we have the medulla. Medulla, which is an important center of activity for the autonomic nervous system, and which contains the respiratory center, most important. The respiratory center is located, the respiratory center, BP center, they are located in the medulla, and this medulla is continued posteriorly as the spinal cord. This is continued posteriorly as the spinal cord, passing through the hole at the base of the skull that is called foramen magnum. So, the brain is located safely inside the skull or the brain box, and it comes out, the medulla part of it comes out as the spinal cord to pass along the vertebral column. Let us devote some time to the cerebral hemispheres. The cerebral hemispheres, or telencephalon, they are the largest part of the brain, and cerebral hemispheres. There are two lobes. In fact, there is a division in the center that divides the cerebrum into two halves, left and right. The peculiarity is that the left cerebral hemisphere, that partition is called Sylvian fissure. So, this Sylvian fissure divides the cerebrum into a left and a right cerebral hemisphere. And the peculiarity is that the left cerebral hemisphere controls the right, and the right cerebral hemispheres controls the left. And connecting the two, however, coordinating the function of the two, are a band of nerve fibers inside the cerebral hemisphere that is called corpus callosum. Corpus callosum is a nervous band characteristic of mammalian brains. This corpus callosum, it's a nervous band joining the two left and right cerebral hemispheres, bringing about a coordination in their controlling functions. Now, each left and right half of the cerebrum consists of 4 lobes. So, left and right, and each left and right cerebral hemisphere consists of 4 lobes. What are these 4 lobes? Frontal, parietal, temporal, and occipital. So, each cerebral hemisphere consists of 4 lobes: frontal, parietal, occipital, and temporal. Temporal on the sides. And the cerebral hemispheres বন্ধ ଆଜି ଆଉ ଆଜି ଯାହା ସରିଲାନି ସରି ହଁ।